said, I'm Matt Fuller. I work at Ultragenyx, and what I've been focusing on for about the last 6-7 years is gene therapy for a variety of rare diseases. And so I just wanted to make sure I said a couple things before we get going. One is a member of the scientific advisory board, I want you to know that I'm here to be able to help you however I can. So my email is at the bottom here, mfuller@Ultragenyx. Feel free to reach out whenever you want. I'm happy to help how I can. If I can't help, I'm happy to try to connect you with someone that can help. Because I know that there are a lot of scientific questions that, you look on the internet, it's really hard to find answers. The other thing that I wanted to say was that I always advise patient groups to really diversify their treatment options. And so I've told several people here, the work that Caroline showed earlier with the ASOs, I think is incredibly promising. And I know, the people that run Mozzie, and I think they're wonderful. And I have a lot of faith in that, but sometimes things just don't work, no matter how great the ideas and no matter how great the people working on it are. And so I think it's always important to think about all the different options you have and have a couple different horses in the race at the same time, just in case something doesn't work out, because there have been times where patient groups have put all their eggs in one basket, it doesn't work, and then you've lost 3, 5, 7 years. And so that's why I think it's important to have this conversation, talk about what might work, what might not work, and then also as technology improves, have those conversations again. Because sometimes things that were not possible 2 years ago are possible now. And a good example is when I met Christine at the Rare Boot Camp, I said, CRISPR basically is a pipe dream right now. It will not work. 2 years later, there's almost an approved drug. And so sometimes things move incredibly fast, and it's important for us to just really keep up with what's going on, reevaluate. So today I'm going to go through 3 topics. One is just the foundational biology of gene therapy. And yesterday, Charlotte gave a really wonderful introduction on the biology. So I don't have to spend a lot of time here, but what I do want to do is just focus on what's critical for understanding gene therapy. Then I'll go through the different gene therapy treatment types. And throughout there, I will give at least my perspective on what I think is important to consider for the treatment of CHD2 by any of these gene therapy modalities. So if we just first sort of look at what is the basic architecture of a gene or DNA, and I think, do I have a pointer somehow? Let's see. So the simplest way to think about it is if you take one of your cells, doesn't matter what cell it is, and you zoom in, you see a chromosome. That's where your DNA is. And someone said yesterday, right, we have 23 of them, they come in pairs, and they're all compacted. And they're compacted into these histones with nucleosomes. And then when you zoom in a little bit further, a unit of these are called a gene. And so a gene is a basic genetic unit that encodes a function of some sort. And genes are located in specific sections of DNA on specific chromosomes. In the case of CHD2, it's located on chromosome 15 in a location that's called 15q26.1. And if you zoom in a little bit more on the gene, you'll see that it's encoded by nucleotides. And I know, again, that was discussed yesterday, but this DNA nucleotide alphabet has 4 letters, A, G, T, and C. And I meant to say earlier, I am happy to share these slides so you can take as many pictures as you want, but I'm also happy to give them to you so you can have a better view of them. And so you have this alphabet, and then if you kind of zoom back out and you look at the gene, each of these genes encode some sort of overt function. And these functions are often in the place of proteins. And I know we've talked a lot about proteins. And so in the next slide, I'll kind of give an example of what a protein really is. And really the reason we're here, right, is that alterations in genes can, but they do not always cause dysfunction. And so that's an important thing to think about. And so again, if you kind of zoom in on the cell, you see the DNA. Our DNA encodes for about 20,000 genes. And these have very discrete cellular functions. And these functions keep us alive. They keep our cells happy, they keep our bodies happy, and that's what's required for our day-to-day existence. And these functions are facilitated by pathways that Gemma and Heather and Charlotte talked about, which is going from your DNA, from your gene, to a message, which is the RNA, and then finally to the protein, which is the molecule that actually does the action that your body needs. And so some examples of protein functions: processing of metabolites, muscle contraction, replicating your DNA so you can make more cells, moving one protein to another part of the cell where it needs to be, activating or inhibiting other proteins. And so if you can think about really anything that your body needs to do, there is probably a protein that is involved in making that happen. And so in gene therapy, what we're seeking to do is replace or repair those very specific pathways and protein molecules when their typical function is not being provided. And what I'll show you next is that you can do this in a variety of ways, just depending on what kind of mutation you have. That's why I wanted to walk quickly through the different kinds of DNA mutations that exist, because this really helps you decide what kind of gene therapy might be best for what you're trying to treat. And so if you see up here on the top left, this is sort of, normal. If we were just to take, one example of DNA to RNA to amino acids. And so, you have 4 amino acids here. And an example of this as a sentence is the cat ate the rat. Now, something that all of us in this room have are something called silent mutations, or sort of variants that really don't change the meaning of the sentence. And so one example of that is the cat ate the rat, with cat being spelled with a K. So it's a little bit different, but we all know what it means. And so your body can tolerate that. And that's really all over your genome. When we start getting into trouble is when we start having either substitutions. So in this case, the hat ate the rat. Now the sentence doesn't make sense. If you insert something and you insert an E here, now we don't even have words. So up here we had sort of the sentence meaning change. Now we have a sentence that doesn't even make sense. It looks like another language. And you can have the same thing if you make a deletion. If you take something out, and it looks like it just makes absolutely no sense. And so understanding the kind of mutation that you're trying to fix really helps inform the kind of gene therapy that might be usable in a situation. Okay. So if we move on to sort of the really most basic definition of gene therapy, it is the treatment or prevention of a genetic disease by introducing genetic material that you expect to provide a necessary function, usually in the form of a protein. And again, if you just think about what I showed you in the beginning, we have a gene that's usually what's identified as having some sort of abnormality that we're trying to fix. And then you're trying to replace the protein that that gene encodes for. And that's what makes it gene therapy, is that we're focused on the gene, and we're focused either on changing this gene or replacing this gene. And the hope is that this will be permanent, that this is a one-time thing. I will say there are caveats to that I'm happy to discuss later, but that's the hope, is that it's a one-time thing. There are 3 really just basic approaches for gene therapy if you just think about them conceptually. One of them is adding back, addition. So if you have a protein that's not working, maybe you can just add back a functional or a wild type copy of the mutated or missing gene. And I have a note down here at the bottom. I don't know if you guys hear wild type a lot, either from doctors or from scientists, but if you do hear it, what that's referring to is sort of the natural or common version. And this is used to describe viruses, protein, DNA, all kinds of different things. So just in case you encounter that. So you can add back. Let's say you have a gene that is being overexpressed or expressing something very strongly that is not working with other proteins. You can inhibit that. You can inactivate it, or you can knock it out. Those all basically mean the same thing. And that is, again, to sort of inhibit a mutated or overexpressed gene that is functioning improperly. So you either have nothing and you're trying to add back, or you have too much of something that's not working and you're trying to get rid of it. And then the last thing, and this is really what has been revolutionized with CRISPR that I know we all read about in New York Times and stuff, is being able to edit. And so if you think about that picture I showed you, where you go to the chromosome, you go to the gene, and then you go to the DNA, you can actually go to the very specific letter or series of letters and change them back in a permanent way. And that's really where the future is going, but there's a lot that needs to be figured out to make this work. Okay, so how do we actually do this? These are the 3 most common tools for administering gene therapy. One of them is called adeno-associated virus, or AAV, and that contains both the delivery vehicle, which is the viral capsid, which is a protein shell that protects the virus, and then it has the cargo inside. And so you can see here, so this is the AAV, this is the shell is in green, and then the blue part is the actual gene therapy that will go into the cell and provide the missing function. Another option is a lentivirus. So it's just another type of virus. You can see that down here. Again, it has a protein shell called a capsid that protects the genome, but also helps it get to the appropriate cell that it needs to go to. And then it has the cargo inside. So again, that's the DNA that encodes the function that you're trying to replace, the gene you're trying to inhibit or activate. And then the last one is a gene editing complex. So that means CRISPR in some flavor, which I'll talk about in a second. And so that is a cargo that must be coupled with a delivery vehicle. And so you can use AAV, you can use a lentivirus, and there are other things you can use as well. That is one of the things that makes gene editing a bit more tricky, is that it's rather large, and getting it intact to the place that you want to go to can be sort of tricky. And finally, as I mentioned earlier, this is a new technology. So for some things, and I'll talk about this later, it seems to be working. For other things, we're starting to get there. And I just wanted to mention the last thing, because I know that I talked to someone about this earlier. Let me see how much time here. mRNA is technically not gene therapy because we're not replacing the gene, but sometimes people think of it as a gene therapy or genetic therapy. You deliver those by lipid nanoparticles, and I'll talk about mRNA in a second, but what I would say is I don't think it's a good solution for CHD2 as the technology exists right now. Okay, so let's go a little bit deeper into the main tools of AAV. So the first one that I discussed was adeno-associated virus. So what's good about it? It's ideal for in vivo delivery, which means you can put this directly into a patient. Now, that being said, because CHD2 primarily affects the brain and other parts of the nervous system, we're not very good at getting AAV there, and it often requires a direct injection. So that's something that really has to be considered. This is ideal for gene replacement. It can be used for CRISPR, but one of the challenges that I'll talk about in a second is that you're limited on how much can fit into that protein shell that you have up here. It's only so big. And you have many capsid choices, which means you can try to get to different target tissues, and people are putting a lot of work and money into this. And the final advantage, and this is really important, is that a lot of academic and industry manufacturing experience exists. Because it doesn't do you any good if you can design this in a lab, put it in a mouse, show that it works, and you can't make it. Or if you do make it, it's like $500 million a dose, and that will never happen, right? So that's a really important key advantage of AAV. Now, the challenges, and I should note, everything I'm going to show you has pros and cons, and just has to be weighed out. It's a small genome size, so that limits what can be put into that protein shell, because again, it's only so big, and I'll comment on CHD2 here in a minute. Even though we have a lot of manufacturing experience, it's expensive. And I'll talk about at the end how much these drugs cost. But you have a limitation of your single dose. So if it doesn't work the first time, it's really difficult to give it again. And there's also immune reactions that are not well understood. For lentivirus, it's good for ex vivo delivery, which means we take patient cells, we treat them with this virus, and once they're good, we put them back in the person. And so that's a really good option for like blood diseases and things like that. This is really not a good option as the technology stands for CHD2. Thing that is better about it, though, is that you can put more into it because it's bigger. You can put 9 kilobases into it, which would be good for CHD2 considering that gene is 5.5 kb. But again, the delivery technology just is not good right now for what we're trying to do. But then CRISPR, what can you do with CRISPR? Now, in the beginning, what you could do with CRISPR is cut things out. But people have been working on this for many years now, and you can do quite a few sort of tricky things. And so if you look up here, you can knock out a gene that was its original function. You can now put a gene in. You can replace a gene into a different part of the genome. You can edit the bases. So as I said, you can go to the specific part of that sentence that's not right and fix it. And then there's a new technology called prime editing, where you can actually put in really big parts of the sequence. So if it's not just, one word like I showed you, and it's maybe a paragraph, prime editing allows you now to fix that paragraph. And as I mentioned, the delivery is a little bit difficult. That's still being figured out. And this works ex vivo. This works for blood and things that you can easily take out and isolate. And we really have to figure out how to do it in a person as they're living. One thing that is important to realize, though, is the targeting strategy must be tailored to a specific sequence. So looking at what people have talked about CHD2, what Gemma and Heather showed the first day, there are mutations all along that gene. And so what we have to do is you kind of pick the most, prevalent mutation, and you make a target for that. Because otherwise you would have to make a drug for every single thing, and all of those would have to be approved drugs. It'd be very difficult. So that is one of the limitations here, is that you probably can't treat everything with one unless you do prime editing and you change a whole big part. Okay, so mRNA, as I said, it's not really gene therapy, it's genetic therapy. It is ideal for large proteins, large genes. The problem is it has to be redosed often, weekly to monthly. So that's fine if you can give it systemically in an IV or something like that. It's not okay if you're trying to deliver it directly to the brain. And right now that would require direct administration. The other thing I would say is that these LNPs over here, these are the delivery vehicles, are actually quite toxic. They're not well tolerated by mice, and they're sort of tolerated by people. And they go to the liver really well, but not anywhere else. So right now, I don't think mRNA is a good option, but I do think, again, we should always keep things in the back of your mind and revisit, because maybe in 2 years, this technology has completely changed and it will be worth looking at again. Okay, so now that I've sort of given you the very basics of gene therapy, what I wanted to do was go through each of the modalities that I think would make sense, and tell you what makes something a good candidate, and then give you some sort of, at least my insights on what I think could be workable for CHD2. So to make a good candidate for gene therapy, again, keep in mind that, I should say, by the way, I've made this capsid look gigantic. It's one of the smallest viruses. It's like 20 nanometers in size. There's only a few smaller than this. So it has a very, very limited capacity, which is only about 5 kilobases. So that's 5,000 nucleotides. CHD2 alone, the coding part is 5,487 base pairs. So you can see just the gene doesn't fit. And there are additional sequences that you have to put in there to make it go into the shell, to turn it on. This is a turning on signal. This is the turning off signal. And so, you have to have all of those things in there. So as it stands, AAV is not a good fit. But what I'm going to talk about is some of the new technology that's coming out that might make this tractable in the future. Another thing that's really important to think about is, and this is something I mentioned for the mRNA, is can your delivery vehicle infect and transfer genetic material to the target cells that are important for correcting the disease? And this is why some of the basic research going on right now, both in dishes and in the mouse, are so critically important, because we don't always know what cells in the brain or in the liver or anywhere else are really important for correcting that disease. It might be neurons, it might be glial cells, it might be astrocytes. And so we have to have really good basic science so we can understand if we are getting this to the appropriate cells to make a therapeutic difference. Then, are the target cells amenable gene therapy? Meaning, once you get this there, are the cells going to accept it and make it work? And I think one of the things that we have to discuss, and I have this later, is we know that from the chaser biology that's been shown from Igor's lab, very elegantly, is that having high CHD2 is also associated with the disease phenotype. And often these diseases in the central nervous system have what we call a Goldilocks scenario. If it's too low, that's not good. If it's too high, it's not good. And if you're putting something into a person permanently, you have to get that right. You only have one chance. That is one of the advantages of the ASO, by the way, is that you can titrate that over time. If you have too much, you can bring it down with your dose. Okay, so what is required for AAV to work for CHD2? As I mentioned, the gene is about 5,500 base pairs. That's about 1,000 base pairs too large, not even counting the stuff that you need to turn it on and turn it off. So this requires using some unique approaches that have existed for a while, but are becoming a little better, a little more tractable. And so you have one called a dual vector approach, where you actually give two AAVs. Oops. And then what happens is you have the DNA split in half, and then they come together and form one large DNA. They're doing this right now for Duchenne muscular dystrophy. It works pretty well in mice. It hasn't been tried in humans, but it's one of those things I think is the technology gets better, it's worth looking into. There's another similar but different technology called inteins. And basically, instead of relying on the DNA to do this, you have the proteins have two pieces that know how to stick together. And so they're also doing this in Duchenne muscular dystrophy. And the dystrophin gene, I should mention, is like 70 kb or something crazy. And they're able to get the entire gene to go together in a mouse. So these technologies really are getting better and better. And then the last thing is identifying a mini gene of CHD2 and testing its function. And so again, the work going on in Gemma, Heather, and Igor's labs are helpful. And I'll explain to you in a second what I mean by a mini gene. But again, I just want to make sure that everyone is aware that these approaches are low efficiency right now. And anytime you put something in the brain, sometimes they have even lower efficiency. And so we have to work to get these, to be in a better place. So in part two, we need to have careful regulation of CHD2. I think that's pretty clear to all of us now. One of the things I was talking to Caroline about last night was that I'm wondering if we can use some of that biology that they've found to put the natural regulatory elements of CHD2 into our gene therapy. And then you could have Chaser modulate that in a person. The other thing is you'd have to use a low strength promoter to make sure you don't have your CHD2 levels too high. And this is why if we were doing this, let's say in a company setting, we'd probably make 20 different vectors when we were starting out, because we would need to test all of this biology. And so I mentioned a mini gene earlier. And so what I mean by that is you actually take out some of the pieces of the protein that you can do without. And so the example of this has been done for Duchenne muscular dystrophy. So I don't have the exact size up here, but it's huge. It takes 24 hours for your body to make the RNA. It's very, very large. And what people have done successfully is they've cut out pieces of DMD or dystrophin, and they've made a microdystrophin or a mini dystrophin, and these are still functional. Now, they're not as good as the full protein, but they're functional, and they provide a level of function that is therapeutically beneficial. And so one of the things that I think really is worth doing is taking all the data that exists and trying to make a CHD2 mini gene, because then you can avoid trans-splicing and inteins, and maybe you can fit it all into one. And that becomes a much better option for treatment. And I would just mention again, all the work that's being done that we've heard about, today, or yesterday and the day before, we're really on the track for getting a mini gene made. Okay, so what makes a good candidate for inhibition gene therapy? What you need is a well understood target. And this can be a direct target. So maybe the protein itself is too high, and so you can directly say, turn that down. Or it can be indirect. Maybe that protein interacts with something, and so you can affect the thing it's interacting with. But what's really critical is having that target really well understood. Because not only is it important for the therapy to be beneficial, but for the FDA to approve it, it's going to have to be clear biology. And it's the same sort of issue is, can you get to the right cell, and will it work on that cell? Okay, so what makes a good candidate for CRISPR editing? I'm going to say the same thing. It's really clear biology, which is important. You need a clearly definable, editable region. I'm not actually sure editable is a word, but that's what I'm going to use here. So usually in genes that have a disease, there is a hot spot of mutation, and there'll be a region, it might be a certain size, but there's a distribution in the patient population. So one example I can tell you of a success for CRISPR is that Editas has been working on Leber's congenital amaurosis, and there's a very common mutation in intron 26. And so what they did is they've used CRISPR in the eye to cut out that region. And I want to say it was like 90% effective. Now, the downside of that, though, is even though it is a highly represented mutation, it's only 10% of the population. So that is, again, one of the downsides of CRISPR, is that there's going to be a mix of the population that can receive it. Then the question of, is the gene truncated or is it a mutation? If it's truncated if it's deleted, there's not really anything to edit. But if it's been shortened or mutated, then you have something to edit. So you have to really think about, what does the biology look like? Okay, and what is required for the addition of CHD2 via gene editing? And it's pretty similar, right? Identification of clinically relevant editable regions to correct. And one of the things that I think might be really helpful is a new technology called prime editing, because this allows for gene replacement in very large corrections. Because as we've seen in the last few days, there are hotspots, right, in the helicase domain and some of the other domains. And it could be that you could prime edit a specific domain. And if you could do that, then you might really have something that's working. But here are the challenges, because it's always important to weigh the pros and cons. It's a new modality, and as I mentioned, CRISPR's only been successful ex vivo. You have to be able to deliver this to the appropriate cells and a large enough number of cells to make a therapeutic difference. If we can only hit 10% of cells, that might not be enough. It might be enough. It's different for every disease, and that's something that has to be worked out by the basic and the clinical scientists. They'll help us figure that therapeutic window out. Base editing has never been used successfully in humans, but again, that's getting better and better. I told you that 2 years ago, I said that this was nowhere near anything, and now there's an almost approved drug. In prime editing, you can insert up to about 10 kilobases. So it's in the range of what you would need to insert a functional copy of CHD2. We just need the technology to catch up with being able to really do this in a person. And I think the clear advantage, right, is the ability to directly fix the underlying genetic cause. You're actually editing the direct cause of the disease. So that would be incredibly powerful, because that would be a one and done kind of treatment. Okay, now that we've gone through all of that, I just want to leave you with the fact that there are gene therapy success stories and sort of some key takeaways. So there have been over 1,100 gene therapy trials across the 3 most common delivery vehicles that I discussed. And there are 4 approved AAV products. And I think someone mentioned Zolgensma the other day. This is one of the most impressive ones. It's for spinal muscular atrophy. And so that disease causes the children that have it to not be able, they have no muscle tone, and they usually die by not being able to breathe. And so the children that have been treated with this are now walking, breathing on their own. It's just absolutely amazing. And so, as I mentioned, right, there's a lot of things you have to get right for this to work, but if you can get it to work, the success is pretty incredible. The downside is, I think you can see the costs up here, $500,000, $2 million, $3 million. And that's something we can talk about later if you're interested. There's a reason the costs are high, and some of them make sense and some of them make absolutely no sense. But I'll be happy to chat with you about any of that. Our approved Linty products, and then I mentioned there's something that's being submitted to the FDA for approval right now that's a CRISPR product, and that's expected to be approved by the end of this year. And so what I hope this does is give you some hope that although there's a lot of work and there's a lot of science that needs to be done, this has been done for other diseases. It is possible. It just takes a lot of work. Okay. I don't have to convince you that gene therapy is a compelling therapeutic modality at this time, but it's important to know that it has the potential to halt, treat, or cure disease with a one-time treatment. What's good about it is we now have broad experience in global clinical trials. And so the FDA understands what we're doing. They understand what we're trying to get approved. So it's a little bit easier in that way now. There's increased awareness and acceptance because of these approvals and because of compelling late-stage clinical results, especially again in the case of SMA. When you see kids that used to, pass at 2.5 years that are now walking, it's kind of hard for the FDA to say this isn't working. You just have to show that it's working for what you're trying to treat. And also the regulators and payers are becoming more familiar with the therapeutic paradigm. So again, we can talk about the costs later. But I think it's important to really focus on why the things that were successful were successful. There was solid basic clinical science. We understood the disease. We understood what happened when we put the gene back. We understood why it worked. There was high unmet medical need. We had good choices of the cells that we were going to, the tissues we were going to. And we understood how to make these things, and that's all getting better. There are limitations though. It's expensive to make. It's not always easy to go to a new target, and there are safety concerns. So in the case of Zolgensma, even though I told you it was amazing, and it was, there were 2 children that passed away during the clinical trial because of heart problems, because a very high dose of AAV9 did not work in their hearts. And so there's always, pros and cons to everything. And that's a real safety concern that the physician should be making you aware of. Okay, so I think I'm right at time. Key takeaways: gene therapy has the potential to halt, treat, or cure disease, and there are successful FDA-approved examples with recombinant AAV, with lentiviral vectors, and hopefully by the end of the year with CRISPR. But the success is critically dependent upon solid basic clinical science, a targetable cell or tissue target, and the ability to make the vector. And I should mention, we can talk about this another time, it's not a given that you can just make these things. So we've been working on Duchenne muscular dystrophy for 2 years, and we have experience making 6 or 7 of these things for clinical trials, and every single one is a new sort of personality of its own. Every single one has to be figured out on its own. So the ability to manufacture is not always a given. As I mentioned at the very beginning, every strategy and delivery vehicle has pros and cons, and we just want to match those to the biology of the disease and the target cells. And I think we should have continued conversations as the technology improves. And finally, manufacturing and reimbursement are expensive. We're working on abilities to improve manufacturing and reduce costs. And the hope is, as more of these gene therapies are approved and as more and more regulators and payers are familiar with this, hopefully that leads to reduced costs. But I can tell you that, especially for SMA, the children who need it, get it. I've not heard of anyone being turned away because they don't have $2 million in the bank. So I do think this is a thing that over time will get worked out. And I think it's everything.